So I'm going to talk to you about tools for personal and population genome interpretation developed in Python. Actually, they're also developed in other languages, but most of the tools that I'm going to talk to you about are explicitly with Python. Just to give you a brief introduction, most of the presentation that I'm presenting here today is on GitHub. I will also upload to my Python notebook and some data sets that I talked to you about during this talk. So feel free to download, and if you have any questions, just send me a message. So brief introduction about me. This is my first time at a Python conference, so take it a bit easy on me. This was highly recommended by friends. Olga actually suggested that I should join you guys and give an introduction as to with respect to what I work on. I am currently a graduate student at the University of Oxford. My main interests include the development of statistical and computational methods for genetic studies of common diseases and developing tools. Outside of that, I also like traveling and dancing. So if any of you guys want to dance at the after party, feel free to interrupt me. Right now, as you may well know, we're currently living in one of the scientific revolutions. So in the past century, we lived in the scientific revolution of modern physics. And I think it's fair to say that this century, or this decade at least, we're living the scientific revolution of genomics. And one of the reasons why this has happened is largely due to the catalysis by the drop in the cost of DNA sequencing. So you see this plot here, right when I was graduating from MIT, the cost of sequencing a human genome was about over $100,000 for an entire genome. And a year before that, it was about $10 million to sequence an entire human genome. So it made it impractical, actually, to do anything with that data. 2 years, from 2008 and 2010, I spent a lot of time working at the Broad Institute. What I was doing at that time, during the huge drop in the cost of DNA sequencing, was developing computational methods, statistical methods, algorithms for the analysis of the raw sequence data that was being generated at that time. And as you can probably speculate, there was a lot of data being generated at that time that related to DNA sequencing. And we had no clue at the time what to do with those data sets. First you identify that there's a whole bunch of errors, and then you need to figure out how you're going to model those errors and improve the ability to make any inferences from that data. During that time period, I decided that one of the things that I wanted to do with my life was probably travel a bit more and also to get a PhD. So I decided to go to the University of Oxford and went to England. And right now, the past 4 years, what I've been trying to do is just trying to finish up my PhD and finish some projects that I worked on that related to taking advantage of the property that lots of genomes were being sequenced during that time period. And what I wanted to do was to try to tie that in with disease interpretation and any health-related trait interpretation. So what's the main challenge? As many of you guys may guess, the main challenge of getting a slew of data is that you don't have any tools or methods to analyze the data. So you have to innovate and think about new ways of processing the data and tying it to the problems at hand. So as I mentioned before, one of the topics that we're trying to figure out how to make inferences from the data was how to process the raw reads being generated by the sequencing technologies to obtaining the best genetic variation, reporting the best genetic variation to perform research. So this is a very arduous research and engineering problem. So this is largely has been tackled from 2008 to 2014. There's still a lot of room for innovation and improvements. As you can see, I've only checked about 2 out of the 3 check marks that it say So right now, one of the rooms for improvement include being able to provide fully phased genomes, saying basically this is, for any given individual, this is the genotype from position 1 to position, the final position of the chromosome, for the maternal inherited DNA and for the paternal inherited DNA. Structural variant detection is actually one area where there's still a lot of room for improvement. The reason is that we've only been limited to short read technologies. And in addition to that, right now, most of the field has been focusing on germline mutations or inherited mutations, whereas it's clear that somatic mutations do play a large role in some disease predisposition, including cancer, but not exclusively cancer as well. So the tools that I'm presenting have nothing to do with this. Actually, there's some nice tools available that I participated in developing during that time frame, including the Genome Analysis Toolkit and this program called Syzygy, which was written in Python. What I'm going to talk to you about is this next stage, which is how to tie in genetic variation or genetic information to personal and population genome interpretation. And this, I would say, if I were to give a check mark out of 3, I would give it half a check mark. I think there's still a lot of room for innovation, still a lot of room for thinking about new ways of analyzing these data sets and ways of how to make inferences about the role of genetics and its contribution to disease predisposition, and also other related traits that probably have not been measured at the moment in any genetic study. So one of the nice discussions I had before the talk was about wearable technologies, and just the prospect of being able to tie those two together is something that's extremely exciting and that hasn't been done. I mean, being able to say, well, this is how physical activity and genetics ties together, that's just something that just seems mind-boggling that it could be done in a very near time frame. So the tool that most of the work that I present today is largely concentrated or developed on top of this toolkit called PLINK-seq. So here's a picture of Sean Purcell. Sean Purcell is actually here in New York City in Mount Sinai. He was previously at MGH and at Harvard Medical School. Sean Purcell actually has developed other widely used tools in genetics, including PLINK. PLINK has over 7,000 citations, and it's fair to say that it largely made genome-wide association studies possible. These are studies that, I would say from 2006 to 2000, or currently, are being carried out to tie common genetic variation and related health traits. So PLINK-seq is developed so that we can be able to tie in genome sequence data or DNA sequence data to health data. The other core developers of PLINK-seq include folks like Menachem, Doug Rutherford, myself, and Brett at Harvard. These two guys are actually here in New York City. So if anyone is interested in reading more about what PLINK-seq is and what is available in terms of tools, this is the link, this is the webpage. It uses a VCF as a primary input. The focus is on the analysis of rare genetic variation, but not exclusive to rare genetic variation. It allows for extensible meta-information on locus, genotypes, and individuals to be integrated. It is also bundled with reference databases that can be directly intersected with one's own data. And I'll show you some ways that can be done. And there's a command line R and a Python library. The Python library, this is a Python package that I have developed with extensions to the PLINK-seq library with Jeff Hammerbacher. He actually wrote a nice snippet of code for Python binding for some key functions in Python. And then what I did was to take that and extend it so that we can start developing additional tools and take advantages of all the aspects that PLINK-seq, of all the methods that PLINK-seq has to offer. So just to give an overview of what PLINK-seq, the world of PLINK-seq is like, there's the command line interface, there's the R library, an Exxon browser, and a Python library. These attach to a core library. which works on top of variation data and any auxiliary data, including phenotypic information, genetic information, sequence information, or any reference databases that you may think of. Just to give you an example of how we may use this toolkit or the pyPLINK binding is, for example, here is a snapshot of 23andMe raw data. So I ended up getting, I ended up ordering a kit from 23andMe, purchased it for $99, and ended up getting back 500,000 markers genotyped across the human genome. And one of the things that you can do with that data, or that 23andMe can do for you, is to provide ancestry inference. I think now they're not allowed to give you back any health related information back due to FDA regulation. But they're allowed to provide the data for you for a raw data download, and you as a user investigator, as a researcher, can go ahead and start processing that data and making inferences of your genotype. So this is what the data from 23andMe looks like. This is just a flat file, and here you'll see RSIDs, which are identifiers for genetic markers in the genome. The chromosome that that RSID is located on, the genomic position, and the genotype that was called by the genotyping technology. There's a nice script available to turn this into a VCF, and as you recall, for pyPLINK, it works on top of VCF. So you guys can go ahead and download that script if you end up ordering or purchasing from 23andMe or from any other genotyping platform, and convert it to a VCF file. So a VCF file is the variant call format file. This is a pretty standard file format file for reporting or reporting genetic data. So here is just an example of what this looks like. There's some nice documentation as to what the variant call format, how the variant call format should look like. And here are examples of commands as to how to work with PLINK in terms of setting up a project, loading the VCF. So this is just an example of me loading my own VCF from 23andMe, and I've uploaded about 922,000 genetic variants that were available for the genotype data. In addition, I can start working in Python and working with pyPLINK to be able to attach the databases that are available as part of PLINK. So you can import pyPLINK, set the project that you're going to be working on. For example, I'm working on the project that contains my genetic data. I can print the summary of the databases that are available. So here are example of databases that are available, that are external databases that are available from other projects. For example, the locus database contains information about the transcript sets that are available in the human genome. And I'll describe a little bit more how that's relevant. There's the sequence database, which basically tells you this is what the positions, or what the human genome looks like. The reference database, which tells you who has, for example, genetic variants at any position of interest. And there's my personal VCF, which I've uploaded to the project. So as I've mentioned before, one of the things that you'd want to do if you're going to interpret what's going on with your genotypes, or if you have any interest in learning about the genetic variation in your genome, one of the things that you'd want to do is to work with these auxiliary databases. So here's an example of attaching a locus database. So once we print a summary of the locus database, we can see that there's several groups available of information. These are basically reference transcript sets that are available in the human genome, and the idea is that you'd be able to use this downstream for analysis and interpretation of DNA sequence variants. So here are 4 examples of reference transcript sets that are available. It's RefSeq, CCDS, GenCode, and Ensemble. In addition to that, another auxiliary database that's available is a summary of the human genome sequence. and that basically tells you the length of the chromosomes, and also if you want to read or find out what the flanking sequence is of a genetic variant, it's fairly easy to do. So here's an example of how we can use those auxiliary databases along with your genotype level data to do DNA sequence variant annotation in Python. So you first load a locus set. So here I'm just going to load refseq. So I've done pyPLINK annotate load refseq. Then, for example, this is just an example of a variant that's on the 23andMe array. You can also iterate over all the variants that exist in the 23andMe array and also annotate them separately or in conjunction. And here is just an example of how you may be able to do that. So you say give a call to pyPLINK, just do pyPLINK.annotate. You give it the chromosome, the genomic position of the variant, the reference allele, the alternate allele. This basically tells it to annotate it. And this is just an empty string, which can be used for other purposes. Like, for example, if you want to find out what gene does that annotation map to. So here you can see that this particular variant is a nonsense mutation or a stop gain mutation. These are, in the human genome, or for every individual, we carry about 100 of these nonsense or loss of function mutations. So these are mutations where that gene partially functions. So you have one copy that works and another copy that doesn't work. So you can call yourself a human knockout version for that copy of the gene, or human knockdown, I would say, to be more precise. And what you can see is that that once we fetch the genotype level data for that particular mutation, so here is a mask. So these masks, I'll talk a bit more about in the next slide, are extremely useful for fetching data from your project. So what I've done here is to fetch the variant level data for this particular variant. And what I've done is to find out what the genotype is for the individual that's in that project. And what you can see is that 0 codes that the genotype is reference homozygous, which means that you don't have the mutation. 1 means that you are heterozygous, which means that you have one copy of the mutation or the variant. And 2 means that you're homozygous for the mutation, meaning that you have 2 copies, meaning that you're likely to have a complete knockout version of that gene. So here I can say that I am heterozygous for this variant, and because of the ability to be able to annotate the variant, I'm also able to say that that for this particular gene, I'm a human knockdown version for gene X. And there's a lot of interest in medicine or in pharma right now for figuring out what human individuals who carry knockout versions of these genes look like, or what their phenotypes are like. And the reason is because some of them may have translational implications. For example, there's a nice story of PCSK9 where if you look at individuals who have knockout versions of PCSK9, they have extremely low LDL levels. And in fact, that ends up translating to having strong protection for coronary heart disease. And when you develop a drug that mimics that function, you actually have a similar impact on individuals who are on statin lowering therapy. So beyond statin lowering therapy, it has a protective effect, being able to knock down PCSK9. So there's a lot of interest in pharma right now in being able to interpret how the phenotypes of individuals who carry knockdown versions for any particular gene look like. So as I've said before, masks are ways where you can easily filter the data with mask syntax. There's a nice documentation on the PLINK website about how you can use the masks. And pyPLINK, you can easily use the masks within any of the calls to pyPLINK by just including it within the quotes. So this is used for extracting information from the various databases that are available. So here's an example of where we fetch the variant data for this particular mutation. But you can also say, for example, just give me the variant level data for this particular gene. So if I'm interested in gene equals PCSK9, I can write gene equals PCSK9. And for the individuals that are in my project, I can find out what the variants are in that database, what genotypes are in that database, and I can also link that to the phenotypes. So other applications of masks, as I've said, you can subset individuals in a project, obtain phenotypes for only certain individuals. You can subset variants based on some quality control filters. You can subset variants based on population-based calculations. So for example, if you have a population of individuals, one of the things that geneticists like to look at is whether the variant is in Hardy-Weinberg equilibrium. And the reason is that that also gives you some sense as to whether or not you actually should care much about that variant, or maybe there's some genotyping errors that end up leading to the property that they are out of Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium is just a calculation for telling you how many copies of each genotype you would expect to see in a population. So this also easily allows inclusion of additional filters. So if you want to exclude criteria for including genes, variants, individuals, pathways, or networks, you can do that. As I've said before, one of the things that I have been doing with the pyPLINK library is to develop my own statistical methods development. So instead of worrying about how to develop infrastructure for working with genetic data, I can save some time and just worry about statistical methods development or ways of interpreting the data. And in particular for working with population scale genomes. So this is a tool or a program developed in Python that I call Mamba, which is for tackling problems in medical genomics. So here, this Python package, what it does is mostly to be able to make inferences when you have population scale level genomes data. So it's focused on genetic association, statistical modeling, and as a project to understand the consequences of these knockout mutations, developing methods for allele-specific expression as well. So just an example of the types of methods that are developed in pyPLINK include this statistical test, which we call C-alpha MRP. And the idea is that with this framework, statistical framework, you'll be able to analyze genome sequence data with high-dimensional phenotypes. So if you have, for example, a population biobank that has multiple health-related measurements collected, and you want to be able to tie the genetics to those phenotypes, you'd be able to do the association, at the level of association, at least with this approach. If you're looking at multiple disease conditions sequenced, and you have a set of related controls, then you'd be able to do the association analysis with this method as well. In addition to that, there's also beyond association, one of the modules that's implemented in Mamba includes the ability to provide estimations of genetic variants that are driving the association and what phenotypes are driving the association. So it's not sufficient to just say that a gene is associated to a whole bunch of phenotypes. You also want to know which mutations are giving rise to the signal and which phenotypes are giving rise to the signal. So this is a Markov chain Monte Carlo algorithm that basically you can see from this. It's fairly easy to run on the data. If you have phenotypes, genotypes, and the number of clusters that you'd want to analyze, then you can run this simple command after integrating with pyPLINK, and it'll give you back some idea as to what the mutations are doing. So this is a representation of about 10 mutations in a gene, simulated mutations in the gene. Here, you can see that there's 3 clusters modeled. So the MCMC algorithm, what it does is to learn the clusters. And here it says, well, cluster 0, we're not expecting an effect for cluster 0 across the 4 traits that were simulated. For cluster 1, we've learned that these are the types of effects that are observed in the data. And for cluster 2, these are the effects that are observed across the 4 different traits. And here you get estimates of the posterior probability that each of the variants contributes to either of the 3 clusters. So here, for example, given that I have simulated the effects, I know what's the truth. I can see that for these 3 variants, the MCMC algorithm actually learns that these 3 mutations impact the 4 traits in a way that's described by cluster 1. Here for these 4 variants, at least for these 3 variants, I'm extremely confident that these 3 variants impact the 4 traits by the estimates that are described by cluster 0, or the null cluster. And here you see that there's quite some uncertainty to it. And then you can interpret what the next 3 set of variants give you. So this is approaches for working with population scale genomes. It's fairly easy to run if you're working with pyPLINK and if you're also working with Mamba. One thing that we hope to do very soon is to provide a lot of documentation about these approaches, and we'll do is to make some data sets publicly available as well. So if anyone's interested in working with these data sets or getting your hands dirty and developing your own tools for interpreting personal and population scale genomes, then feel free to work on it. So if you have an interest in developing genomics applications with Python, feel free to email me, and we can chat about it later as well. Okay?